Okay, so on this first session, we would have our first speaker. Ms. Obeid is right here. Okay, good. So let me say something about Ms. Obeid. Dr. Obeid, I'm sorry. You can see that I'm taking Jenny's role now. Okay, so Dr. Obeid is professor of medicine at SUNY Stony Brook. She received her MD in 1983 from the American University in Beirut, Lebanon. She did her internal medicine residency program at Duke and a fellowship in endocrinology and geriatrics and some work at Duke at the same time. She rose through the ranks at Duke to become associate professor of medicine and cell biology. And in 1998, she was recruited to the Medical University of South Carolina in Charleston, where she served as the Boyle Professor of Medicine in the Division of Geriatrics, and as Professor of Biochemistry and Molecular Biology and Staff Physician at the Charleston Veterans Affairs Hospital in 2012. She was recruited to become Professor of Medicine and Dean of Research at Stony Brook University in 2017. Is that correct? 2012, okay. So she launched her research career by studying the role of signaling lipids in cell stress. So she's going to talk more about how these lipids stress out cells. So that's a very good topic. We're very happy to have Dr. Obeid, and her topic today is going to be on bioreactive sphingolipids, how they play a key role in cancer metabolism. So thank you, Dr. Obeid, for coming.
Good morning, everyone, and thank you so much for the kind introduction. I know Jenny's on her way. They texted me, and it was really bad traffic this morning. So we came the night before and stayed at a bed and breakfast, which was really nice and smart idea to do. Anyway, good morning. This is a beautiful building. It really is, and I'm so happy to be here. And I know I'm happy to see Dr. Ian Taylor, who is the person who recruited me to South Carolina from Duke. We were friends at Duke, and then, so good friend and great person. Okay, so the talk today is going to be about bioactive sphingolipid metabolism, and I changed the title to The Plot Thickens to make it more on cancer, but I sent my slides before I made many changes, so at any rate, basically, the talk is going to be about these lipids that we work on that are very important in cell regulation, and we've uncovered a really interesting role for them in cancer. People don't think of lipids as associated with cancer, but over the last 2 decades or more now, I think this pathway has become really well studied in cancer. So what I'm going to talk about is this bioactive lipid pathway. And it looks a bit scary when you first look at it, but the basic, what's amazing about this pathway is it starts by the condensation of a very basic amino acid that's very important in diet and also important in cancer, as I tell you in a bit, and palmityl-CoA, which is a fatty acid that is, again, very important in diet and in many, many related diseases. So these two molecules, palmityl-CoA and serine, start condensing together to form the backbone of sphingolipids, which is this basic sphingoid, 3-ketosphingolene, which is then quickly reduced to form dihydrosphingosine, and then it's acylated, so another fatty acid is added to make dihydroceramide. And it's an interesting pathway, but the bottom line is you get to this molecule, ceramide, which is a very bioactive molecule, and we implicated it in cell death, in cancer cell death, more than 20 years ago now, and it's really opened, us and others opened a really nice field to study this pathway in cancer. So ceramide in cells has one of two fates, one of several fates. It either becomes phosphorylated to make another bioactive lipid, and the blue ones, all the blue molecules are bioactive, meaning they on their own have biologic activity. So sphingosine phosphate is a pro-inflammatory lipid, whereas ceramide is a pro-cell death lipid. Ceramide can also be made into sphingomyelin in cells, which is 10% of all cell membranes have sphingomyelin, is sphingomyelin, and it's a very important molecule because when it's hydrolyzed back to ceramide, and the enzymes that regulate its hydrolysis are implicated again in cell death. Ceramide can also be modified by glycosylation to make glycolipids, which are again very important molecules that sit in cell membranes and form places where receptors can interact, or many of the complex sphingolipids can be important in immune cell responses. Ceramide can also be broken down by a ceramidase. There are many of these enzymes, and to make a bioactive molecule called sphingosine. And these sphingolipids are named because with the sphingo thing in the front, meaning they were enigmatic lipids, and it's based on the sphinx, people didn't know what these lipids did. So I think we're slowly solving the enigma of the sphinx by understanding these molecules. Sphingosine can then be phosphorylated to make by sphingosine kinase, a very, very important and highly regulated enzyme, to make sphingosine 1-phosphate, which is, again, itself a very bioactive molecule. And it has receptors in cells, G protein coupled receptors, 6 of them. And when it binds its receptors, it's really actually 5 of them. And there are other receptors that bind it, but we don't know if they are specifically binding. They're different family of receptors. But so these are very bioactive, and I'll talk more about them in a bit. But what I'm going to do first is outline where we are interested. We've been really interested in ceramide and sphingosine phosphate. These molecules because of their bioactive activity, and so we are very interested in the enzymes that make them and break them. And today I'm going to spend most of the time talking about these two enzymes, sphingosine kinase, and a new enzyme pathway we discovered that takes ceramide and sequesters it, so it's out of cell death pathways. And I'll try my best to explain this because I know the audience is very diverse. Okay, next slide. So why these sphingolipids? Again, because of their biologic activity. So I'm going to just show you, they're made by the de novo pathway, as I said, but it's becoming increasingly obvious that cell stress responses induce generation of ceramide in cells, whether it's done by the de novo synthetic pathway or by, before I get to that, whether it's done by the de novo synthetic pathway or by the breakdown of sphingomyelin. So either from de novo synthesis or breakdown of sphingomyelin, you make ceramide. Ceramide, we and others have shown, is a critical lipid to induce cell senescence, differentiation, programmed cell death, and cell cycle arrest. On the flip side, ceramide, as I said, is broken down by ceramidases to make sphingosine, which is then phosphorylated to make sphingosine phosphate by this enzyme sphingosine kinase. And the product sphingosine phosphate acts very differently than the first product, ceramide. It induces cell proliferation, mitogenesis, inflammation, migration, angiogenesis, and protects cells from apoptosis. So it's immediately obvious that the balance between those two pathways and these two molecules is really crucial. And so that's why we've been very interested in how this pathway is regulated, and that's what I'm going to spend time about. So I will start by ceramide, discussing ceramide. Ceramide is synthesized in cells, as I told you, by the de novo synthetic pathway. Interestingly, there are 6 enzymes that make ceramide in cells. And they make ceramides, they're called CERS, ceramide synthases, 1 through 6, and they make ceramides of different chain lengths. And I'll illustrate this by this picture. So you can see that the head group is the same in all of them, but the side chains are longer. They're C14 side chain when they're acylated, or C16, C18, which means the length of the fatty acid that acylates. So these long fatty acids are added on to the backbone of the sphingoid to make these longer chain sphingolipids. And there are even longer ones that I'm not talking about today that are found in skin, and also the molecule can be regulated in other ways. So what's interesting about these lipids is they are bioactive, but they also are structural components of membranes. As you see, they're like highly hydrophobic molecules. So why are there so many ceramide synthases in cells? So one of our questions was to try and solve this problem. So a very talented postdoc who is now actually a junior faculty and just got his first RO1 with the 6th percentile, so I'm very proud of him, is John Senkal, who actually came up with this idea. He decided that to understand how these different enzymes work, he was going to immunoprecipitate, tag all of them, all the 6, tag them with a flag tag, like a light bulb, and then bring that down and see what other proteins associate, and do they associate differently or the same, so in order to understand their function. So what he showed is, these are the different ceramide synthases, and he showed the red is proteins that associate with each individual one using a proteomic approach. So we studied that using proteomic analysis. And what he found was that one particular protein associates with all of the different ceramide synthases, meaning that it must be important in this pathway. Plus, he found many proteins that associate with different ones, and he's taking these into his work as he moves forward with his own grant. But what's remarkable here is this protein is called fatty acyl-CoA synthetase. So it basically makes a fatty acid. And so this is an enzyme that makes another long-chain fatty acid. They're called very long-chain fatty acid synthetases. So we wondered what the role of this protein is and why is it associating with all these ceramide synthases. And this just proves that when you bring down the protein, each of your proteins, that's each one of the ceramide synthases, you immunoprecipitate it, and then you do a Western blot, you can see that they actually all interact at different strengths. So this is a biochemical proof that they are interacting. So what about this protein? So this protein is called long-chain fatty acyl synthetase. Now this, it's a long-chain fatty acyl synthetase, so it makes long-chain fatty acids. And it's an important de novo lipid synthesis, so really critical in making lipids in cells. And it functions in fatty acid uptake, so it's important to take up fatty acids in cells. And it's also found to be highly expressed in cancers, remarkably, a lipid-making protein, highly expressed in cancer. So that got our light bulb got us more excited. And its downregulation in cells, if you knock it down, can induce cancer cell death in lung and glioma, colon, and other cells. Sorry, I forgot, this is out of the frame. But basically, it forms this reaction. A fatty acid plus ATP makes acyl AMP and a phosphate, and then using coenzyme A, it adds and makes an acyl-CoA. So it makes the fatty acid ready to be incorporated into other lipids. So, and what we hypothesized is, since ceramide is made of a sphingoid that's acylated with a fatty acid, we thought this was what it was here for, interacting with ceramide synthase to make ceramide. But we were wrong, and I will not go into great detail why we were wrong, because this is published in a very nice paper by John. He's first author on it, called In Cell Metabolism, and he basically showed through what he did was he modulated this enzyme to see if it changes ceramide levels, and it was doing the opposite. When he knocked it out, it made less ceramide, not more. So we were surprised. So then when we looked more carefully, and I'm making a long story short, but the publication is very accessible for you, and you can understand more. But I just want to tell you how we moved it forward. But what John found that, in fact, this enzyme, ACSL, is interacting with ceramide synthase to add another fatty acid to ceramide. So this is the basic molecule of ceramide, and it's adding another fatty acid to make this huge lipid, which is somewhat similar to triglycerides, because triglycerides that you find in fat are made with 3 fatty acid chains. So this was very intriguing when we made this discovery, and we proved it by knocking out the different enzymes. So you don't have to take my word for it, but we did prove it, and we're pretty confident that this is what's happening. And now we were wondering, so what is the protein? So here you're adding the ACSL, which is the fatty acid CoA synthetase, with the ceramide synthase. But then, but then there needs to be a protein that actually does the transfer, the fatty acid transfer. And so we looked for this protein, and we found that, in fact, to find this enzyme, we looked at what happens with triglyceride, how are triglycerides formed. So from the chemical structure, that to make diglycerides in cells, you have this diacylglycerol, which has it's an alcohol with 2 fatty acids attached and a 3rd hydroxy group. And then if you add this enzyme, diglyceride acyl transferase, which is a critical enzyme in fatty acid in triglyceride synthesis. This is the main enzyme in making triglycerides in cells. So if you add that fatty acids, you make triacylglycerols, which are like all these lipids that we have in our body and that are possibly regulated by our diet. So we asked the question then, if this looks similar, does actually this enzyme, the same enzyme, diacylglycerol acyl transferase, take ceramide and add an acyl chain? And indeed it did. And this was a very important discovery because no one had implicated this protein except in triglyceride synthesis. So we showed it's actually making acyl ceramide, which is really interesting and different than anyone knew about the function of this protein. And then we wondered, why is it making this, and how is that happening? And so what we looked is that acyl ceramide, when was reduced in 60%, if you have, oops, I'm sorry, this went fast. So if you have the DCAT knockout mice, the enzyme that makes this acyl ceramide, if you knock it out in the mice, actually ceramides the mice are actually eventually are very sick because they can't make ceramides in their skin, and they become very permeable, and they cannot be well because they're losing water through their skin, because ceramides form the permeability barrier. So that told us that this enzyme, when you knock it out, not only does it affect glycerolipid synthesis and triglycerides, but it does affect ceramide synthesis. So we felt we were on the right track. And then in yeast, in Saccharomyces cerevisiae, this enzyme was responsible to make acyl ceramide in that species. So that was another hint for us. So then actually we can say, and again, this is in John's paper, we can say, and we did knockout and manipulation to prove, and then we showed by biochemical studies as well that de novo ceramide synthesized by ceramide synthases that I told you about, the 6 enzymes, all interact with this ACSL5, and they also interact with diacylglycerol acyl transferase to form this acyl ceramide. So this is a new pathway in lipid synthesis that we discovered and we're very excited about. And now I'm going to tell you our newer data that's unpublished about the implications of this. So from that paper, the conclusion was that fatty acid come into ACSL. They interact with ceramide synthesis and with DGAT, diacylglycerol acyl transferase, to make acyl ceramide, and that this acyl ceramide is incorporated into lipid droplets. And this is the proof here that if you transfect cells with the ACSL and the ceramide synthase, you can see here that you're getting lipid droplets in cells, and if you do higher magnification, you see the interaction is at the lipid droplet interface. Similarly, if you transfect with DGAT2 and ceramide synthase, you also see that they interact. And when it's yellow, meaning that the green and red are interacting, that they are, again, at the lipid droplet interface and in the lipid droplets. So what's happening is when these three proteins come together, they make this molecule, acyl ceramide, and it goes into lipid droplets in cells. And what's really interesting is that lipid droplets in cells are thought to be people didn't understand what they were. They thought they're just lipid accumulation. But now we know that lipid droplets in cells are very biologically active. They're metabolized, they're regulated, and they're very critical in triglycerides in cells. But we've implicated them here also in harboring ceramide and in taking ceramide to acyl ceramide and incorporating it into lipid droplets. So what are the implications to this? So what we did then, we did a mouse model, and here what we did was we take mice and we feed them either a regular diet or a high-fat diet. And on a regular diet, we take their livers, and their livers are healthy. But if you feed them a high-fat diet, mostly oleate, a fatty acid, they have fatty liver. And then if we inhibit this enzyme, DGAT2, from making the acyl ceramide, in fact, they are protected from fatty liver, and they lose the acyl ceramide, and they make more ceramide. So that was interesting from 2 points of view. First, they regulated cell death, the ceramides that were generated, and also they protected from liver disease. So then we wanted to move this work. Obviously, we work on cancer. We're very interested in cancer. So one question we asked, which people had looked at this enzyme, the acylglycerol acyl synthetase, but no one had ever wondered if it had anything to do with cancer. So first thing we did was we looked at the cancer genome database, and we found, to our surprise, that many cancers overexpress the enzyme DGAT is highly amplified in these cancers. Using the cancer genome database. And I outlined ovarian cancer, breast cancer, pancreatic, prostate, and pancreatic. These are all cancers known to have lipid droplets in them. And there are data in the literature to say cancers that have lipid droplets in them are much more aggressive. So we were interested in breast cancer in our group. And so we also looked at the other DGAT, which is DGAT1, and this is even more amplified. And in all of them, the first amplification is from a xenograft cell line of breast cancer. But you can see in pancreatic, ovarian, again, prostate, and breast that they're highly amplified. So we were interested in what's the implication of this amplification. Is that indicating that in cancer these enzymes are working, and what are they doing? And also when we looked at KM plotter to see survival, we find that patients who have high expression of DGAT have a worse survival. So if they express this enzyme and they make a lot of lipid droplets, they have worse survival. So that got us interested in trying to manipulate this enzyme to see if we can rescue cells from cancer. So what we did is, and I'm not showing you a lot of data because there's another subtopic I want to talk about as well, but I just want to give you this highlight information that we took several breast cancer cell lines. These are SKBR3, and they are a HER2 cell line, HER2 positive cell line. We took MDA-MB231, this is a mistake, cell lines, and these are P50 triple negative breast cancer cell line. And we took MCF7 cells, which are epithelial breast cancer cells that are P53, normal for P53. But in every case, what we showed is if you take the inhibitor of DGAT, so the cells are treated with the inhibitor of DGAT either at different concentrations, 0, 25, 50, and 100 micromolar in every case. And you see the white bars that not much happens to the cells when they're treated with the inhibitor alone. But if you add a chemotherapeutic agent, then you are really synergizing cell death, which to us, we know because we measured, what happens is when you treat with this inhibitor, you no longer make acyl ceramide in cells, so you free up this acyl ceramide and it goes into ceramide. And ceramide accumulates, and when ceramide accumulates, it acts in synergy with chemotherapy, which we have shown in the past that ceramide and chemotherapy increases ceramide in cells many years ago. But when you add the inhibitor with the chemotherapy, then you are really synergizing and increasing cancer cell death. So for us, this was exciting because it's happening in many breast cancer cell lines and in response to different inducers, cisplatin or doxorubicin. So this for us means that we can enhance cell death in breast cancer cells that are resistant. And what is remarkable is that these inhibitors for DGAT synthesis are already in the clinic because they are being used to inhibit triglycerides in the blood, and they are novel inhibitors of triglyceridemias. They are somewhat toxic because they leave a lot of fat in your gut and they cause GI symptoms, but that can be probably studied better. And the cisplatin and other chemotherapeutic agents are in the clinic. So if you put the two together in a clinical trial, we expect that maybe we will enhance cancer cell death. So that's one of our goals in the near future. And we're working with Dr. Alison Stopik to potentially put a protocol for cancers, like a phase 1. Is that called a phase 1 or a phase something? Anyway. 4 minutes. Okay, so I took too long on this. So I'm going to go into the next part of my talk. Oops, I'm sorry. So the conclusions from this part is ceramide can be converted to acyl ceramide and stored in lipid droplets, leading to resistance to cancer cell death. And ACSL1 and DGAT interact with ceramide synthases. DGATs are amplified in cancer, and they are important for, if we inhibit them, it's important for cancer cell death. So these are important for implications to cancer treatment. And we're very interested in the mechanisms, and that's the next phase of this work, including hopefully that we would do some more animal and then clinical research. So as I told you, this pathway, we discussed it. One of the other parts of so we've added to this pathway a new addition, which is that ceramide is acylated to acyl ceramide. So I think the pathway is getting bigger and more interesting. So then I wanted to, in the last few minutes, tell you that mostly sphingolipids are made, like I said, by serine and palmitoyl-CoA, but occasionally, if you don't have serine, or you deprive cells of serine, cells can use alanine, a different amino acid, or they can use different fatty acids. But what happens in that case is, so I'm going to skip all this and just tell you. So this is the enzyme Sphingosine Kinase that I told you about, and this is just a scheme to show you that it's very biologically active. That these are all the products, these are all the pathways that it's involved in, and it's clearly involved in cell death pathways. So, and it is proteolyzed in response to DNA damage, one way to protect. So what we did is, when you look in cancer cells synthesize serine at a higher rate, and they require, because they have endogenous some serine, but they also have to make serine because it's not enough for them. And so there was this paper in the literature from the Maddox et al. That showed that when you deprive cells of serine, they are protected and they reprogram cells in cancer. So we were interested because we had seen that our enzyme, Sphingosine Kinase 1, was degraded in DNA damage in a p53-dependent manner. So when we took p53 out, it was no longer degraded. So because of their paper, we wanted to see what happens in our cells. And we found that indeed, when we take colon cancer cells and treat them deprive them of serine, Sphingosine kinase gets degraded. The enzyme that is like very potent in cancer becomes degraded. And we showed that this happens in wild-type colon cancer and in p53 null, but you can see the enzyme is getting lost. So, and that this is mediated by a protease, because when we inhibit proteolysis, we inhibit this breakdown. And I'm sorry that I have to rush through this, but that's when we inhibit this enzyme, sphingosine accumulates, and sphingosine phosphate, the very bioactive molecule, goes down because we're inhibiting the enzyme. I just want to get to the part to tell you that when you form these non-canonical sphingolipids, if you remove serine from cells and use alanine, you form non-canonical sphingolipids that are missing this important hydroxy group that you see in all canonical sphingolipids. And that is really important because this hydroxy group is what gets phosphorylated to make sphingosine phosphate and make bioactive lipids. So when you're missing it, what happens is the sphingoid is no longer able to be phosphorylated by sphingosine kinase, this important enzyme. And apparently, it's been known that when you have things that bind or interact with that enzyme and are inactive, they lead to proteolysis of the enzyme and its breakdown. So we think we know the mechanism why when we deprive cells of serine, sphingosine kinase is proteolized, and it leads to accumulation of sphingosine and to deoxysphingosine, which is this non-canonical sphingolipid. And here I'm showing the accumulation of these non-canonical sphingolipids when you deprive cells of serine. They really accumulate, and they are toxic to the enzyme. So they basically, when they accumulate, when you treat cells exogenously with them, they break the enzyme down. So if you're looking at, and then if you inhibit proteolysis, the enzyme is no longer degraded. So that tells you that they are important in regulating the enzyme. And so we were really interested in this because we wanted to see if now when we knock down this enzyme by this pathway, what does it do to serine synthesis? And to make a long story short, because of time, and because my timer just went off, I'm going to show you that the final scheme and close. So what we showed in this is that serine, if it's not there, not available, cells can use alanine, and they will make this non-canonical toxic sphingolipid called deoxysphingolipid, which leads to proteolysis of sphingosine kinase. So you can no longer make sphingosine phosphate, and the cells become produce reactive oxygen species, inhibit mitochondrial function, and lead to synthesis of serine. So there is a pathway that these molecules, and we're not sure how yet it happens, but they drive PHGDH, which is a key enzyme in serine synthesis. So when you inhibit this pathway, you modulate cells to induce serine synthesis. So I just will close here by saying that we've two important pathways, fatty acids and amino acids, are really critical in regulation of sphingolipids, and by looking at mechanisms, we found that they were important in regulating cell death in cancer. So both have implications to clinical studies, and now I don't show you, but we did some studies in mice where we deprive them of serine. If they have a tumor, the tumors shrink. And so we're trying to study what happens to the sphingoids. So we have modalities that are important in diet that we hopefully can use in fighting cancer. And I will stop by acknowledging my coworkers. So my lab group is the following people, and I talked today mostly about John Senkal's work and about J.P. Truman's work. And we have very good collaborators. We have a lipid cancer lab. We all collaborate. And Yusuf Hanoun is always great for advice. So is Dr. Mao and Dr. Luberto. Dr. Snyder helped us with the fatty acid work. Her interest is in fatty acids in the diet. And Jeff Gernon helped us in the serine work. And our core facilities are really crucial. And then my funding sources. And I really appreciate your time. And I'm sorry I went over. Thank you.
Thank you very much, Dr. Obeid. Very nice talk. Now we know that lipids can stress out cells. And when lipids stress out cells, bad things happen. So let's keep that in mind.